angiogenesis was the first argument in my life. I started the angiogenesis work at the National Cancer Institute many, many years ago. It was 88. And we started with Suramin, a very old antiparasitic drug, which was developed as a potential anti-angiogenic drug because of its ability to bind the so-called fibroblast and PDGF growth factors. At the time, VEGF was not known, but and then people, very famous people like Napoleone Ferrara came and discovered VEGF and all the biology of angiogenesis. Of course, here in the room we have Bob Kerbel, the guru of angiogenesis, I should say, and so I'm very much impressed to be here today, and thank you again to involve me in this very challenging experience. As a background, I am a clinical pharmacologist. I like to interact with oncology people, and my work, or the work of my group, is focusing on different aspects of tumor biology. One colleague of mine is working very well and very hard in angiogenesis research, and he joined the laboratory of Bob Kerbel several years ago. We are moving also on different fields like the circulating tumor DNA. We are trying to develop the biomarker story more closely, and so I was very much impressed about the presentations. So I'd like to say something about the clinical experience in gastric cancer and the positioning of anti-angiogenesis drugs in Europe. Dr. Shah presented the pivotal trials on Ramucirumab and bevacizumab in particular, and provided a very, very nice and very good description, very detailed description of those trials. So I'm just showing you a couple of slides to connect to the considerations made by Dr. Shah. And I will talk more on the regulatory side, just to explain you what the European Medicines Agency did for the new anti-angiogenesis drug Ramucirumab, and for the indication of Ramucirumab for the treatment of advanced metastatic gastric cancer. We published a review paper very recently. It was published in this month, in 2015. And it was just an occasion to review the story about the angiogenesis inhibitors in gastric cancer. So we put in this review all the information available. We made a review of the clinical information available in the treatment of gastric cancer and gastroesophageal junction carcinomas with anti-angiogenesis drugs. I'd like to put some colors in the presentation, otherwise it will be gray presentation with no colors compared to the other presentations. But you know very well the story about angiogenesis. Today was a very productive meeting, very interesting meeting. Many information. I really enjoyed to be here today. And we have many targets. We have many ligands. We have many receptors. We have many potential pharmacological targets for anti-angiogenesis drugs. And for this reason, we have developed so many drugs and so many new molecules for anti-angiogenesis inhibition. Probably today we have more than 20 anti-angiogenesis inhibitors. And the design of the new drugs spans from, the classic antibodies that block the ligand of the anti-angiogenesis treatment. We have the new antibody targeting the VEGF receptor 2. We have a huge amount of multi-target tyrosine kinase inhibitors. They differ between them for very few characteristics because they all target many kinases. The difference between these drugs is more on the concentration needed to inhibit the target, but they are multi-kinase inhibitors, so they target many kinases inside the cells, and the list is very wide. We have many drugs. We have drugs like cediranib, more specific on VEGF receptors, but also cediranib. If you look at the IC50 of the drug towards the different targets in the endothelial and cancer cells, you will see that also cediranib, even if it is more specific for VEGF receptors, also inhibits a number of kinases inside the cell. And we have this new approach, is the peptibody. This drug is very active and is a strong inhibitor of angiopoietin, is the trebananib, as a complicated name. All these drugs have very complicated name. I like the TSU68, it's more simple. But I'm sure after this, it will be given a very complex name, I'm sure. So, I mean, the targets are many. There is redundancy in the control of angiogenesis. We have many drugs targeting different biological targets in endothelial cells and in tumor cells. But today, the results in some cases are encouraging, in others discouraging. And the problem is we do not know exactly what is the reason why one drug is successful in one disease and is not successful in another. Or at least we are trying to understand. We have seen beautiful presentations this morning and today and in the afternoon. We're trying to identify the biomarker, but the biomarkers is elusive in angiogenesis research. So the only approach today is to conduct clinical trials in unselected patient population, at least for a biomarker. The only selection we make is based on previous treatment, which is sometimes very bad because previous treatment can select a more aggressive tumor phenotype. So, I mean, the difficulty in developing new anti-angiogenesis agent is obvious. We have clinical trials. Dr. Shah already explained you the main results of the AVAGAS, the AVATAR, the REGARD, the RAINBOW trial. We can see here that the line affects significantly the overall response rate, the progression-free survival, and the overall survival of patients, because if you go from an overall response rate of 37, 46 in the AVAGAS trial in first line, you drop, you see here, to smaller numbers at least, because if the line advances, the biology of the tumor changes. And we know that a pre-treated tumor develops a resistance mechanism or survival mechanisms that are able to overcome the cytotoxic or pharmacologic effect of the drugs. And we have encouraging results in some trial, very good results, I should say, with Ramucirumab. In particular, the combination of Ramucirumab plus chemotherapy, this is a Paclitaxel-based chemotherapy, is encouraging. We have good numbers in terms of significance in overall response rate, lower in terms of progression-free survival and overall survival. But, I mean, the story, it's interesting. The way is encouraging. And we need to improve our ability to select patients, that's for sure. And that will be a turning point to improve our ability to treat patients with gastric cancer with anti-angiogenesis drugs. We learned interesting things by dividing the ethnic groups and the patients in the clinical trials, enclosing patients with Chinese ethnicity or general population. So we learned many things about the success and limitations of anti-angiogenesis treatment in gastric cancer. We have new drugs, of course, as you can see here. the Tribananib. If the treatment line is first line, the results are better because we have improved overall response rate when we treat patients in first line. But if we go to second or third line, the results, of course, are much poorer. And we will see in the second slide. But unfortunately, in many cases, we don't see a significant difference in terms of overall survival, overall response rate, or progression-free survival. So the numbers here are quite disappointing. Gastric cancer is a very aggressive disease, and in the metastatic setting, the aggressiveness of the disease is obvious, is demonstrated by numbers. Here are additional studies with additional targets and drugs, apatinib, furatinib, orantinib. The difference between these drugs are not dramatic. Those drugs can target VEGF, MET, FGF receptors with different specificity, with different potency. The issue is, I don't think we can discriminate the drugs based on the nanomolar concentration because they are very close, actually. So there should be something more. These drugs target many targets inside the cell. The concentration required to inhibit the target is very low today. We have improved significantly the profile, the pharmacologic profile of these drugs. But again, we have not very encouraging results. As you can see here, if the line of treatment is very late, like third or later, progression-free survival is very reduced. But if you compare at least to placebo, a treatment can increase the progression-free survival and can even increase the overall survival. At least this is good information, is encouraging information. But consider that we are comparing the drug against placebo, which is ethical in third line. We have additional information in second line with furatinib, but as you can see here, numbers are not very encouraging. Orantinib, first line, not very much increase in the clinical parameters. So we have success, we have disappointing results, we have all those things. We need to think about the significance of these results and make a wise selection of the treatment that we are going to propose to the patient. Of course, when talking about the new treatments, Ramucirumab is a very important drug. In Italy, we have a program of compassionate use of Ramucirumab for patients with advanced gastric cancer. And as you can see here, it's very recent an update on the orphan drug designation of Ramucirumab for the treatment of advanced gastric cancer resistant to previous chemotherapy. And this is a document written and released by the European Medicines Agency very recently. So the point is the European Commission, just to talk about the European situation, I'm not talking about science. I mean, the science was very well explained before, but I'm just trying to comment with you and to share the information we have on the present situation in the European Union. As you can see here, the European Commission granted a marketing authorization for Ramucirumab very recently, the 19th December 2014. So each member state is implementing this authorization in different ways. Of course, there will be member countries much faster compared to other. In Italy, as I said before, we have a program of compassionate use for Ramucirumab in patients for the treatment of advanced gastric cancer. Of course, each time we make a decision about the drug, there should be a balance between benefits and risks. And the CHMP, which is the Committee for Medicinal Products for Human Use, decided that in a positive way that Ramucirumab benefits were greater than its risk and recommended the use in the European Union. As you know very well, the main approved indication of Ramucirumab in these patients is for the treatment of adult patients with advanced gastric cancer or gastroesophageal junction adenocarcinoma in combination with Paclitaxel. I think that the choice of Paclitaxel is reasonable. One, because these patients must have disease progression after prior platinum or fluoropyrimidine chemotherapy. So the idea is to move to a combination with a drug, a cytotoxic drug, which has also anti-angiogenic ability. We know very well that Paclitaxel and the other taxanes have an anti-angiogenic effect. And for this reason, I think that the combination of Ramucirumab with Paclitaxel makes sense, or at least is a combination with a cytotoxic drug with some specific characteristics in the inhibition of endothelial cell migration and proliferation. So, I mean, it's a prototypic anti-angiogenic drug, Paclitaxel. As I said before, patients must have disease progression after platinum or fluoropyrimidine chemotherapy. There is also approval for Ramucirumab monotherapy, which is indicated as with the same indication as before. Of course, the benefits of Ramucirumab monotherapy are smaller. We have better results if we consider the combination with the Paclitaxel, and we will make a couple of considerations later. Also in this case, in Europe, patients must have disease progression after platinum and 5-FU or capecitabine combination treatment. And the approval for Ramucirumab monotherapy is in these cases in which we know that the patient is ineligible for combination with Paclitaxel. One potential ineligibility criteria is neuropathy developed before. Of course, the benefit was smaller when Ramucirumab was given alone, but it is considered by CHMP in the European Medicines Agency still a therapeutic option when, as I said before, Paclitaxel is not considered appropriate. The CHMP concluded that the benefit is considered clinically relevant, given, and this is an important point, of course, the poor prognosis in these patients, because we know very well that the prognosis of gastric cancer in these conditions, particularly after chemotherapy, cytotoxic chemotherapy, the prognosis is particularly grim. The safety profile, this is important, but also I will stress additional points later. The safety profile of Ramucirumab is in line with what is expected for other anti-angiogenesis drugs, and again is considered acceptable given the benefits of the medicine. So the conclusions of the CHMP were positive by considering all the results in the of the clinical trials. I wish to stress a point that the European Medicines Agency said when the Ramucirumab was approved, is that there is a successful and satisfactory risk management plan developed to ensure that Ramucirumab is used as safely as possible. I will address this point later. And of course, the agency wants to stress the safety of the treatment. The safety is, of course, an issue. Again, we are talking about patients with short survival, short life expectancy. So the issue of safety is absolutely fundamental. It's necessary to be addressed in a proper way. So this is why the European Medicines Agency put the black triangle on the information on Ramucirumab, because the agency, as for most drugs, I will address also this point later, the agency recommended additional monitoring, safety monitoring for Ramucirumab, and this is to allow quick identification of new safety information or safety issues. Again, the CHMP said that the benefits and the risks for Ramucirumab are reasonable and they are positive, so the balance is in favor of its use in gastric cancer. And also, I mean, the clinical trials demonstrated that the safety profile is what is considered for an anti-angiogenic drug. But again, it's important to do the monitoring within the pharmacovigilance system. What does the black triangle mean? The European Union has introduced this new way of identifying medicines that are being monitored particularly closely, and this is the case of Ramucirumab. Of course, there is a mandatory implementation of carefully monitoring of the medicines once they are placed in the EU market. And this is because, again, in the real-world setting, the information we have about safety can change, because in the real-world setting, we are going to include different patient populations, patients with comorbidities, patients with co-treatments. So for this reason, a black triangle is a note of caution in order to acquire additional safety information before releasing all the information needed to the public. But of course, it does not mean that the medicine is unsafe. It's only a matter of additional monitoring in terms of safety. This strategy of addressing the safety information, and in the case of Ramucirumab, is because, again, as I said before, in the real-life setting, the real life is sometimes quite different from the strict regulation of clinical trials, because, again, in the real-life setting, a larger and more diverse group of patients will use the medicine. So what we need to know is if in the real world the safety profile will change, and if we can use, the drug in a safe way. And this is the reason why the regulatory agency asked additional safety information to be collected within the pharmacovigilance systems. This is a normal procedure for all the medicines authorized since the start of 2011, and in particular for anti-cancer drugs. This is a mandatory requirement for the collection of additional information and to make the use of the drug safer. One strategy used to address the safety issue is to conduct, this is a potential strategy, it's not the only one, but this is a potential strategy is to conduct post-authorization safety studies, the so-called PASS. And the post-authorization safety study is a study related to an authorized medical product we conducted with the aim of identifying, characterizing, or quantifying a safety hazard. This kind of study is aimed to confirm the safety profile of the medicinal product and also to measure the effectiveness of risk management procedures, which is very important for the safe use of all the medicines. As I said before, the objectives of a post-authorization safety study is to quantify the potential risks or identify new risks, just in case there are new safety issues that emerge during the real-life use, to evaluate the risk of the medicinal product in specific patient population for which safety information are limited, particularly patients with renal or hepatic impairment, but not only them. Also to assess patterns of drug utilization and again safety, to add knowledge on the safety of the medicinal product in patients with co-medications. And again, as I said before, there is a strong need of risk minimization activities and plans, because one crucial issue is the safe administration of a drug to the patients. These are the objectives. Of course, the post-authorization safety study can be conducted on a voluntary basis. This is usually recommended by the European Medicines Agency, but also the post-authorization safety study can be performed as an obligation imposed by the authority. And as I put in this slide, in this case, if this is an obligation, this is a condition for the granting of the market authorization. Of course, this is not the case for the Ramucirumab, but I'm talking in general for the new drugs reaching the market. So the could be an obligation and could be a condition to reach the market, to obtain the marketing authorization, or also can be considered as a condition after the granting of marketing authorization if there are concerns about the risk of the authorized medicinal product. So the approach is obligatory or voluntarily, but the issues addressed are the same, the safety profile of the drug. This post-authorization safety study can be done also when the marketing authorization is granted under exceptional circumstances, when the drug gives extraordinary advantage of survival in a specific patient population, has an accelerated approval. But again, also in that case and even more, the post-authorization safety study is absolutely important to address this specific point. On the voluntary basis, studies are required to address the risk management and investigate the eventual safety concerns, and to address in a precise way the risk minimization activities. So for Ramucirumab, we obviously reached a new potential treatment for patients with gastric cancer. Ramucirumab is one of the many anti-angiogenesis drugs tested in gastric cancer, but is one of the few that reached positive results. So in terms of conclusion, I should say that although pivotal randomized clinical trials testing bevacizumab provided the initial disappointing results, there are new data that confirm that multiple mechanisms account for the efficacy of anti-angiogenic inhibitors in patients with gastric cancer. Ramucirumab is a novel opportunity, is a novel drug. There is much interest on the use of this drug in gastric cancer, but not only in gastric cancer, also in other cancers like non-small cell lung cancer. Is a new inhibitor of VEGF receptor 2, and what is very important is the ability of Ramucirumab to provide an advantage in terms of overall response rate, overall survival, and progression-free survival in patients pre-treated with chemotherapy, even alone, better if in combination with Paclitaxel. There are additional results which are interesting on apatinib. There are indications to increase our knowledge within phase 3 trials. But again, it's interesting to show how VEGF receptor 2 is a valuable target for the control of angiogenesis in gastric cancer. And of course, I like to underscore the importance of translational research, we discussed so much this morning on the importance of the better knowledge of the process of angiogenesis in order to identify the tools that are able to stratify patients and identify those patients with higher chance to respond to anti-angiogenesis treatment. Of course, if we throw the drug in a general population, we could have disappointing results. But if we were able to select patients, we will see much better results, of course. But it's very encouraging to see what happens in the case of Ramucirumab and gastric cancer. I mean, in unselected patient population, pre-treated with chemotherapy, so a difficult patient population, the drug is able to provide the clinical improvement in their condition. So the issue of anti-angiogenesis is viable, it's important, is worth to be pursued. And of course, if we will be able to understand more on the intrinsic of acquired resistance, we will be able to improve our treatment. And this is why we need new compounds, but we need more new biomarkers to select patients.